Complex molecules required for life, such as the amino acid arginine, are synthesized by our cells in metabolic pathways where different enzymes catalyze different steps of the pathway. That is the conversion of one intermediate, such as the amino acid ornithine, into citrulline, and the conversion of citrulline into arginine. The identification of the genes that control these catalytic steps in metabolic pathways is an important goal of genetics. And microorganisms like the mold Neurospora crassa are a very useful laboratory model to dissect these pathways, to discover what are the different conversion steps, as well as the genes that catalyze, or the genes that encode the enzymes that catalyze these conversion steps. So in order to discover the genes, usually what is done is that bacteria or the mold Neurospora crassa is grown in on plates, and you can see the plate over here. And wild type Neurospora crassa will grow in a very robust manner, but different mutants can have varying levels of growth. Now, wild type Neurospora crassa are prototrophic, and that means that they can synthesize all the complex molecules if they are given certain salts and a carbon source, such as glucose. And this combination of salts and a carbon source is called minimal medium. And therefore, wild type microorganisms, it could be bacteria, Neurospora crassa, are capable of synthesizing all the complex molecules just starting from minimal medium containing just glucose. However, if you have mutant Neurospora in which one of these genes, let's say gene Z, has been mutated, then they will not be able to synthesize arginine or whatever complex molecule synthetic pathway that we're working on. It won't be able to synthesize this complex molecule starting with just minimal medium, and will need to be provided a supplement. Since the pathway is broken, it'll need to be provided with a supplement in order to grow well on these plates. And such strains, such cells are called auxotrophic, that require supplementation in addition to the minimal medium in order to grow. And so one basic way of discovering genes is to carry out what's known as a mutant screen, where the mold or whichever organism that we happen to be studying is mutagenized using X-rays or other mutagens in order to create mutant strains, and then those strains are tested for growth on minimal medium. And if those strains, a particular strain is not able to grow on minimal medium, but is able to grow when supplemented with some molecule, then you know that one of the genes involved in the biosynthesis of that particular molecule has been mutated in that mutant. For example, the ARG1, ARG2, and ARG3 mutant strains of Neurospora, they cannot grow on minimal medium, but will grow just fine if the minimal medium is supplemented with arginine, and that tells us that all the three mutants are involved in the biosynthesis of arginine. And the goal in such experiments is twofold. One goal is to arrange these compounds or products that you are supplementing minimal medium with in the order of the pathway. And the second goal is to identify which mutant and hence which gene controls which catalytic step in the pathway. So which gene encodes the enzyme that controls the conversion of one particular intermediate to another. So in these data that we have been provided, we have been given the three mutants over here of Neurospora crassa, and these three mutants were grown in three types of medium. You had minimal medium supplemented with arginine, minimal medium supplemented with citrulline, and minimal medium supplemented with ornithine, giving us a total of 9 data points. So we note immediately that if you supplement the medium with arginine, the strains always grow, and that implies that none of the blocks occur after arginine, and therefore arginine must be the last step in the pathway. By a similar set of logic, citrulline is blocked at only one mutant, and therefore it must be the second last step in the pathway, and ornithine must be the second step in the pathway. Now, once we have decided on the order of the intermediates in the pathway, next we have to decide which mutant and hence which gene controls which particular step in the pathway. And in order to do that, we will write the wild type alleles with plus superscripts, and so we'll have arg2 plus and arg3 plus, with the understanding that the wild type allele makes the enzyme, whereas the mutant allele does not make the enzyme. And so now we would like to identify which enzyme is encoded by which wild type allele of these genes. And we note that in the comparison between the minimal medium supplemented with arginine and minimal medium supplemented with citrulline, in the arg3 mutant, even if you supplement the medium with citrulline, and so you're adding a bunch of citrulline over here, you still cannot get growth. Therefore, the pathway must be blocked at this stage in the arg3 mutant, with the implication that the wild type allele of arg3 plus, makes enzyme Z. And so if you mutate it, then there is no enzyme Z, and therefore this pathway is blocked at this stage. And whether you add citrulline or even if you add ornithine in the arg3 mutant, there is no growth because you get stuck at the citrulline stage. The citrulline never gets converted into arginine. Now, we can use a similar set of reasoning, and we can compare the ornithine supplemented medium condition with this citrulline supplemented medium condition for the ARG2 mutant. And here, if you supplement with citrulline, you get growth over here. However, in the ARG2 mutant, when you supplement with ornithine, you do not get growth, with the implication that the ARG2 mutant is blocking the conversion of ornithine to citrulline, and that the ARG2 plus, the wild type allele, must code for enzyme Y. And finally, the ARG1 mutant must be blocking the conversion of the precursor into ornithine, since the ARG1 mutant will grow when supplemented with ornithine or citrulline or arginine. And that is how we determine both the order of the compounds or the intermediates in the pathway, as well as assign which mutant is blocking which part of the pathway. Let's practice working with pathways with an example problem. Here we have been given a pathway where a colorless intermediate is converted into a magenta intermediate by gene P, and then this magenta intermediate is converted into red pigment by an enzyme encoded by gene Q. And we are asked what would happen in a plant that is homozygous for a null mutation of P. And a null mutation means that the allele makes no functional protein. And so if you are homozygous for this null mutation, that means there is absolutely no enzyme to convert the colorless intermediate into the magenta intermediate, and therefore you're blocked at the colorless stage, and so these plants will be colorless. Furthermore, let's write down the genotype of these plants, and we will assume that the genes are unlinked. And so this individual would be little p over little p, and we will assume that they are homozygous for Q, and so they will be big Q over big Q. Next, let's consider what would happen in an individual who is homozygous for a null mutation of Q. And if you have a null mutation of Q and you're homozygous, that means you have no enzyme to convert the magenta intermediate into the red pigment, and therefore this individual is going to be magenta, and their genotype is going to be big P, little q, little q. Third, we are asked what is the phenotype of an individual who is homozygous for null mutations of both P and Q. So their genotype is little p over little p and little q over little q. And evidently, both the steps in the pathway are blocked, and therefore the individual is going to be stuck with the colorless intermediate, and their phenotype is going to be colorless. Okay. And lastly, we are asked what F2 ratio is expected if the first and second plants are crossed and their F1 progeny are selfed. And so our cross is the first individual, little p over little p, big Q over big Q, and we are crossing them to an individual who is big P, little q, little q. In the F1, we will get a dihybrid individual who will be big P, little p, and big Q, little q. And this individual will be making both the enzymes, since they have one big P allele, and they will make the enzyme to convert the colorless intermediate into the magenta intermediate. Similarly, they have a big Q allele, which will make the enzyme to convert the magenta intermediate into the red pigment, and therefore they will end up being red. However, this is not what we are asked. We are asked if this individual is selfed. So we will take this F1 individual and we self them. What will be the proportions of the phenotypes in the F2? Now, this is a dihybrid cross, and we know that we will have four types of genotypes. We will have individuals who are P over dash and Q over dash. We will have individuals who are P over dash, so they will have the dominant phenotype for the first gene and little q over little q, recessive for the second gene. We will have little p, and big Q over dash. So the recessive phenotype for the first gene and the dominant phenotype for the second gene. And finally, we will have individuals who are homozygous recessive for both the traits. Furthermore, we know that in a dihybrid cross, you get a 9:3:1 ratio, and so we have 9/16 of the individuals have the dominant phenotype for both the traits, 3/16 have dominant phenotype for the first trait and the recessive for the second, 3/16 have the recessive phenotype for the first trait and dominant phenotype for the second trait, and 1/16 have the recessive phenotype for both the traits. So next, we need to decide what are the phenotypes of these individuals. So the first individual has the one enzyme making allele for both the genes. So you have a big P, which will convert the colorless intermediate into the magenta intermediate, and you also have a big Q, which will make the enzyme that will convert the magenta intermediate into the red pigment, and therefore this individual is going to be red. The second individual has one big P allele. So they will have enzyme to convert the colorless intermediate into the magenta intermediate. However, they are homozygous for the recessive allele or the mutant allele for the second gene, and therefore they will not make the enzyme that converts the magenta intermediate into the red pigment, and therefore they will be stuck with the magenta intermediate, and they're going to have a magenta color. The third individual is little p over little p. So they'd have no alleles that make the enzyme that converts the colorless intermediate into the magenta intermediate. So they are going to not be able to convert the colorless intermediate into the magenta intermediate. They do have a big Q allele, and they can, in principle, convert the magenta intermediate into the red pigment. However, they will not have any magenta intermediate because they are homozygous for the mutant allele of the first gene, and they will never make magenta intermediate. And therefore, the third individual is going to be colorless. And the last individual is mutant, is homozygous for the mutant alleles of both the genes, and therefore both the steps in the pathway are going to be blocked. However, the second pathway is irrelevant, because if the first step is blocked, then you are stuck in the colorless intermediate, and so this last individual will also be colorless, and therefore we get a 9 is to 4 is to 3 ratio of red is to colorless is to magenta in the F2 progeny if the F1 individuals were selfed.